Okazaki fragments, but we don't want to leave them as fragments. We actually want to attach them to make one nice long strand of DNA when we're done synthesizing. So what we're going to do is we're going to use a specialized enzyme called DNA ligase. Okay. So just so you can see here, template strand. Okay. There's the RNA primer, polymerase 3. Okay. There's an Okazaki fragment. It reaches the next primer, which was most likely the primer that started the leading strand there. So this might be an origin of replication, or it could just be a previously made Okazaki fragment. And then the polymerase is going to actually detach in this particular instance. It's going to attach to the next area where there's a primer. It's going to synthesize DNA up to that primer before it detaches again. Okay. Once the Okazaki fragments are formed, then we're going to have ligase that comes in here and actively binds. First we're going to have a different type of polymerase that replaces all of these primers. And finally, we're going to have ligase come in here, and it's going to stick all the pieces of DNA together. Okay? So, just kind of a laundry list of all the proteins involved in this process. Okay? Helicase we talked about. Single-stranded binding proteins we talked about. Topoisomerase and primase talked about. Here's the big ones that I'm going to go back to this slide and show you one more time again. DNA polymerase 3. Is the enzyme that is going to be responsible for using the parental strand and producing the daughter strands of DNA. DNA polymerase 1 is going to be responsible for coming in and replacing the RNA primers with actual DNA nucleotides. And DNA ligase will join them together. So again, this here, DNA polymerase 3, this here, DNA polymerase 1, this here, ligase. Katie and then Richard.
So the DNA 1 create the Okazaki fragment?
Say that one more time.
Will the DNA 1, not 3, produce the Okazaki fragment?
No. 3 is responsible for both the leading and the lagging strands. So the Okazaki fragments that are made here are made by DNA polymerase 3, just like the leading strand's DNA was. The DNA polymerase 1 is responsible for coming in after these Okazaki fragments have been made and replacing any RNA from those primers with DNA. And then, once we've done that, we're still going to have a break. You'll notice right here, where this DNA meets this primer, okay, or vice versa. So we're going to have ligase come in, and it's going to stick the two together. It's going to join the two pieces of DNA. Richard?
Is there such a thing as a DNA primer?
A DNA primer?
Because you keep referring to these as RNA primers?
These particular primers are indeed RNA, for a very specific reason. Okay. First and foremost, the RNA primers act as kind of a navigation site for DNA polymerase 3. Without RNA primers, DNA polymerase would not know where to bind on the DNA to start replicating and making new DNA. And I think that it does that. I think that our cells have evolved to that point because when you're replicating DNA, you're just going to have a ton of DNA. And it would be very, very difficult for an for a DNA polymerase to recognize where specifically on that DNA to start if you simply had a DNA primer bound to a template strand of DNA and then other DNA primers on the other DNA strand. But an RNA primer, which is going to have uracil and ribose, makes it slightly distinct from DNA. So it makes it a very, very easy, starting point because there are those discrepancies between the DNA and the RNA. Now, you can, I believe, to a degree, make DNA primers, but the majority of polymerases are going to utilize RNA as the primer that they recognize to initiate replication. Okay. And I think when you order from companies, typically you order RNA primers.
Thank you.
Yes?
You said that the ligase, at the very bottom of it, it joins two pieces of what?
DNA together. Okay. So again, polymerase 3 elongates the strand. DNA polymerase 1, and don't you love how they did that again? Because the one y'all are going to want to think is doing all the hard work is polymerase 1, because it comes first. But no, polymerase 3 is used first to make or I guess to synthesize the complementary DNA, polymerase 1 is used to replace the RNA primers with DNA, and then ligase comes in after that and ligates the two pieces of DNA from the Okazaki fragments together. Any other questions, concerns about that? Yes?
Would the RNA primer be pretty much the same thing? I mean, you said that it's replacing it. Number 1 is replacing it. Polymerase 1 is replacing it. But is there really that much that needs to be replaced? Or does it replace the whole thing regardless?
It replaces the entire thing.
But it would be the same thing.
So even if it's an adenine and a cytosine, it's going to replace it.
Okay.
And the reason for that, Richard, is because of the sugar. If you look at the sugar, and here's what you need to know. The reason why we can go back decades now and actually analyze DNA from crime scenes is because DNA as a molecule is one of the most stable molecules that you can find, okay, in terms of nucleic acids. And that comes from the fact that DNA has, for its primary sugar, for its primary sugar, it's going to have what's known as deoxyribose. It looks like that. The only difference between deoxyribose structurally and ribose is that at the 2 carbon, instead of a hydrogen, ribose has a hydroxyl. And that hydroxyl functional group makes ribose just a little bit less stable than deoxyribose. And you see this constantly. If you actually ever work with DNA or RNA, with DNA, you technically are supposed to wear gloves because you have enzymes on your fingertips called nucleases that can chew it up. But even if you choose not to, the molecule still has a certain integrity to it. So it might get chewed up, but some of it might remain. But I'll tell you right now, if you don't wear gloves with RNA, you're not going to see anything when you go to analyze it because it's so, I guess its stability is so jeopardized by the addition of this hydroxyl group that the presence of those enzymes almost immediately results in the molecule being degraded. So, stability-wise, DNA much more stable. That's why we want to replace those primers with DNA primers, or I'm sorry, DNA nucleotides, because that deoxyribose is going to add an extra element of stability to the molecule that otherwise wouldn't be there.
Some of the primers you typically work on in the lab, they're only about like 5 to 6 weeks back.
They can be between 5 to about 30 tops. And usually they're RNA primers, and what they usually wind up doing is they make these hairpin loops on them so that they're a little more stable when they ship. Okay, so use this chart in your book to your advantage. It's going to provide you with a lot of the information that you need just in general on these different types of enzymes. Okay, and here's what the whole process, all going on at the same time at one replication fork looks like. So one last time, I will walk you through this, and I will take it slow. Okay? So initially, helicase is going to unwind the double helix. Remember, we need the origin of replication for that to occur. The helicase starts at the origin of replication and unwinds the double helix. Remember that there's a helicase at this fork and one over here at this fork. Now, as I pull the 2 strands apart, I'm going to start building up stress at either end of the molecule of DNA. So what enzyme am I going to use to alleviate that stress?
Topoisomerase.
Good. So it's not shown here, pictured, or not pictured here. Topoisomerase is alleviating the stress of the molecule. Okay, now next question. How am I going to keep my strands from reassociating? What am I going to add to each of them? A single strand binding protein. Good. So I'm going to add a single-stranded binding protein in multiple places to each of my strands to stabilize them so they don't reassociate with one another. And finally, if I want my DNA polymerase to be able to bind, it needs to recognize a specific sequence so it knows that's where it needs to start replication. So what enzyme do I have to use first? Primase. And what is primase going to make?
RNA primers.
That are recognized by DNA polymerase. So now I've got my primers. I've got a leading strand that's going to be made and a lagging strand. DNA polymerase 3 is going to bind wherever it sees a primer, and it's going to begin to replicate the DNA. And if it's on the leading strand, it's going to make one nice, uniform, long piece of DNA. If it's on the lagging strand, it's going to make these Okazaki fragments. Okay? These short fragments of DNA. Okay? So leading strand, lagging strand. After DNA polymerase 3 has made these Okazaki fragments, DNA polymerase 1 is going to come in and replace the RNA with DNA. Once we are only working with DNA, I'm going to have my leading strand, which again is nice and uniform in length, does not have fragments. I'm going to have these Okazaki fragments that have been converted now to all DNA rather than just RNA and DNA. And ligase is going to come in and stick those fragments together. What parts would people like me to repeat? Yes, Kate?
So it's
The Okazaki fragments are made on the lagging strand. Yes. Sure. So you've got your origin of replication. This is the site that basically initiates replication. So what will happen at the origin is helicases will come in and begin to unwind the double strand of DNA in both directions. So there'll be a helicase moving in this direction to unwind the sequence going towards that fork, and one moving in that direction. Okay? From the origin of replication. You literally are opening up the helix in both directions. As you unwind the helix, if you think of it like string, if you pull string like kite string from the center, the string is actually going to start to bunch up above or below where you're pulling that string apart. We don't want that to happen with the DNA because it can slow down the entire process. So we have another protein that acts, and it's outside of this picture, called topoisomerase. And what it does is it cuts the DNA and reorients it to alleviate the stress that comes with unwinding the strands in both directions. Okay? Yes, Josh?
What happens when the bubbles, like two bubbles
Meet? Okay. So when two bubbles meet, that's a great question. What will happen is the polymerases for both of those are going to actually run into DNA that's already been made. So they're going to fall off. So we're now going to have two DNA strands that are now meeting, just like we do here. So what enzyme do you think is going to come in and stick them together? DNA ligase, right? Good. That's exactly what happens. Okay? So anywhere the DNA fragments meet, whether it's a really long fragment meeting a little short one or two short ones meeting, we're going to have ligase come in and actually adhere those two together, or ligate those two together. Any other questions? Yes, Allison?
You said, I don't know, this was after you talked about primates. You said DNA polymerase 3 will do something.
So primase lays the primers down. Polymerase 3 recognizes those primers as points where it needs to start replication. Okay? So think of these primers as the starting line in a race. Okay? If I just give you an empty track with no labels on it, and I say, okay, Allison, you got to start your race, you're not going to know where to start. But if I paint a big old starting line, I say, okay, Allison, you need to start your race, you're probably going to go up to the starting line, right? That's what these primers do for our polymerase. They're the starting line for replication in terms of polymerase. You don't put those primers down, no replication. Does that make sense? Okay. Any other questions?